So today I'm going to be talking about a mix and match, getting comfortable with the DNA reporting. And we were tasked with talking about what's in a match and how to read a forensic DNA report. So that's what I'll be focusing on. I'll start with a case example, and then we'll go through the information, demonstrative aid that a jury or a trial attorney would see, and then use that to explain what a report looks like with probabilistic genotyping or TrueAllele, and then describe what the discovery material looks like so that when it's all done, you're comfortable with how DNA is reported using statistical interpretation. So in Elmira, New York, which is about 5 hours north of here, in 2011, a man broke into a woman's apartment and he had a knife and he was wearing a bandana. He raped her and he was recognized as Casey Wilson, who is the boyfriend of a friend of the woman. 2 years later, a man broke into a different woman's apartment, and again with a knife, he raped and robbed her. And then he tried to eliminate evidence of the sex crime. And again, Wilson was recognized by his voice and his height. After the second crime, there was a surveillance video that showed a masked man who was wearing purple knit gloves, who was leaving the second victim's apartment, and later on the surveillance video found him back near where Casey Wilson lived. So this is the case of the people of New York versus Casey Wilson. The main evidence of interest were the gloves from this Elmira serial rapist. What he had done with the gloves is he used the gloves to break into the apartment, to do things inside the apartment. After the assailant had raped the woman, he was wearing the gloves when he douched her to try to clean out any evidence of DNA, which of course put more evidence onto the gloves. And then he wore the gloves as he was leaving on his bicycle, and the bicycle and the gloves are found in different areas in a nearby park. There was the hope that evidence found on the glove that was biological from the victim and the assailant could connect Casey Wilson to this crime. This is an example where DNA mixtures are really powerful. If you can find 2 or 3 people on them that bring together a crime, they're really informative forensically. The laboratory in Albany, in New York, analyzed the data, but this was a low template complex mixture with at least 3 people. And due to insufficient genetic information, they couldn't analyze the first sample to get a statistic. And with the second, they had a left glove and a right glove. With the second glove, due to the complexity of genetic information, again, they couldn't make a comparison or get a sample. There was a grand jury coming up in a day or two, and an analyst from the lab contacted the district attorney in Chemung County and said that we might be able to analyze it. So the data was sent to us. Basically, in 24 hours, we got the data, analyzed it, wrote a preliminary report, got it back out. They continued the grand jury hearing, and then we had a complete report by then, the following week, and I went up and he was indicted, and then a year later there was a trial. So what these slides are about to show is pretty much what we presented at that trial when I testified in Elmira, New York. This is the understanding that a forensic scientist or a prosecutor, a defense attorney, or a jury or a judge would have, the way we present in court now about TrueAllele results. This is the people versus Casey Wilson, and this is a computer interpretation of the quantitative DNA evidence. So on the left, we see a cell. The body has trillions of cells, and most of these cells have a nucleus, and inside that nucleus, shown in blue here, are 2 complete copies of the human genome, one inherited from a person's mother, another from a person's father. The genome is basically information. It's text. It's like a book or a hard drive. It's not like proteins that fold up in 3 dimensions. It's text that you read, say, from left to right. On the right side of the screen, you see a chromosome, one of the 23 packages from one copy of the human genome, into which this text is packaged. And if you unravel it and open it up and take a look at a small area shown all the way on the right, there's a double helix, and you can actually, with microscopes, special microscopes now, make out the letters, the A, C, G's, and T's that make up the 4-letter DNA alphabet. So that's what DNA is from this perspective. Yes, it's the blueprint of life, and it grows cells and handles processes and disease, but we're really focused on it now for our purposes in forensics is text. And the sort of text that scientists have been looking at for the last 20 years for genetics, genetic diagnosis, gene discovery, and forensics is the short tandem repeat. Scientists have moved on with other methods of looking at human variation, but in forensics, particularly because there are these databases that already exist and have tens of millions of people in them around the world, the short tandem repeat, or STR, has had quite a long shelf life for a technology. It's based on PCR, which is the amplification mechanism discovered in the 1980s. And let me tell you what a short tandem repeat is all about. In the genetic encyclopedia that we have, shown on the left, is 23 volumes, each volume corresponding to one of the chromosomes. So this is half of the genetic content of a nucleus. The other half would come from another parent. There are scattered throughout this encyclopedia about 100,000 regions that all have the same property. They have a fixed region of DNA, then they have a variable region of DNA that differs between different people in the population. Then there's a fixed region of DNA again. And if we look at that paragraph at one location, or locus, the DNA locus paragraph, the starting text is English. This is not DNA speak, but it's more understandable than ACGAATTA, right? Take me out to the ball game, take me out to the crowd. We're not going to start singing, but that's how it goes. And then the paragraph continues, and somewhere in the middle, in the genetic encyclopedia, there's a word that repeats. It can be 2, 3, 4, 5 letters long. In forensics, it's usually about 4 letters long. This 4-letter word here is root. It goes root, 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 root. In this case, we repeat it 10 times. That's what happens to be in that paragraph in some particular page of the encyclopedia. And then it continues again for the home team, and it ends at the old ball game. So what you have is that depending on the number of times the word root is repeated, that paragraph will change its length. If you count up the number of letters in the paragraph and the words are repeated 10 times, you have whatever the length is, and you have 10 occurrences of root. If root appeared an 11th time, there'd be 4 more letters, and the paragraph would be 4 letters longer. So what scientists do is they measure the length of the paragraph to figure out the number of times the word's repeated. The longer the paragraph, the more times root's repeated. The shorter the paragraph, the fewer times it's repeated. The number of repeats of that 4-letter word is called an allele, or the type, the genetic variant, of that paragraph that's in a particular place in a particular volume of encyclopedia. And the way scientists find it is they basically do a string search. They put in, the way you might do in Google or LexisNexis, the phrase, take me out to the ball game, that's the beginning. At the old ball game, that's the end, both shown in red. And then the chemistry knows how to find everything in between and make millions of copies that are labeled and can be detected on a genetic analyzer. So that's the technology. You're measuring the length of the paragraph, and that length tells you the number of times the middle word is repeated. That's the basis of STRs, or these short words that are repeated in tandem. More importantly for human identification than an allele is the genotype, the pair of alleles that a person inherits from their 2 parents, because that's what identifies a person. In this case, again, opening up a chromosome shown on top and unraveling it and taking a look at one location or locus, there's text there, and there's constant regions on either side. But then when the repeats occur, this picture is showing that perhaps the mother allele in this person on this one chromosome in their cell that's inherited from their mother has a 4-letter DNA word in the ACGT alphabet repeated 10 times. The other allele in the same volume of the encyclopedia, but the encyclopedia inherited from their father, has the identical paragraph, but now there are 12 repeats of that word. And that pairing of 10 repeats and 12 repeats of those 2 alleles is what forms a genotype. And so a person's genotype at one location would be a 1012. Typically with STRs, there are about 15 different values that would occur, and that means 15 times 15 over 2 is around 100. There are about 100 possible genotype values that a person can have at one location. There may be 10 or 20 that are most common. If you multiply those possibilities, 10 times 10 times 10 times 10, or 100 times 100, 15 times, that's where you get the trillion, or 1 followed by 24 zeros, possible genotypes. And in human identification, comparison really isn't made between people, it's made between genotypes, the barcodes that we inherit. And with that number of possibilities, if there's only 7 billion people, which is not even 10 zeros after the 1, but there's a trillion possible genotypes or barcodes, which are 24 zeros after the 1, then there's a much larger space of possible genotypes than there are people. And all the statistics that we talk about are comparing genotype to genotype. The result of this is that within that space of possible genotypes, a person's individual genotype is pretty unique. How is DNA evidence interpreted? This is a general framework. I'm going to describe it for single source DNA. So assume that you don't have a mixture, there's no degradation, everything's perfect. Lots of DNA, either a cheek swab, blood on the floor. It's from one person, lots of DNA. On the left, we see DNA, maybe an evidence item from one person. That arrow that says lab, the lab does extraction and quantification and amplification, separation, detection, and it produces data. At every one of these 15 or so STR locations, there's a signal. And the signal is based on size. The longer the paragraph, the more repeats there were, the larger the allele. So going from left to right, we can read out 8, 9, 10, 11, 12, 13, 14. That's the number of repeats, or the alleles. And what the data signal would look like from clean, pristine, single source data for someone who may have had a 1012 genotype actually in their DNA, one on each chromosome in each cell, there's a signal in the center where there's a signal, a peak at 10. There's a peak signal at 12. We don't see anything at 8 or 9 or 11 or 13. They're roughly balanced because there's an abundant DNA for the experiment and because genotypes come in pairs of alleles. So that's what single source DNA looks like. That's the signal. And then the inference step shown on the right arrow is to determine what that genotype is. Well, with data this simple from one person, out of 100 possibilities of what the genotype value could be, of what those allele pairs can be, the data collapses those 100 possibilities to just one possibility, the 1012. That collapsing of possibilities, the reduction of possibilities by using data, is what information is all about. That's where genetic information is being obtained, and that's what statistics, which we'll get to, match statistics, are all about. In this case, it's quite simple. The 100 possibilities of this location are reduced to a 1012 allele pair. And later on, because this is all done objectively, no one's looking at the answer, a comparison is made. And when a comparison is made to a known genotype with a 1012, then a match statistic can be produced and numbers can be generated. But what I want to talk more about is DNA mixture interpretation, the work that computers can do when there's 2, 3, 4, 5, 6, or 7 contributors to a mixture. You get into 5 or 6 or 7 with handguns and touch DNA. And we see quite a bit of that. But let's stick, for this example, to 2 contributors. So on the left, we have an evidence item that has DNA from, say, 2 individuals. The lab does exactly what it would have done before. It extracts the DNA, it quantifies, it amplifies, and then it separates these labeled fragments out on a genetic analyzer. And now we see evidence data that can contain more than 2 alleles, which is an indicator that there may be more than one person in here. And in this case, we see a 10, 11, and a 12. There's a pattern. The 12 seems to be taller than the 10 and 11, which statistically indicates, doesn't have to be, but indicates that there's more of a 12 allele that's present than of a 10 and 11. And now for the separation step, because that's what computers can do that people really can't, and that's to be able to separate out genotypes into their different contributors in a way that can be used to obtain information. Suppose that we already know for some reason that the first contributor is that same 1012 that we saw before. We want to now establish for the second contributor at this one of 15 locations, what is that second genotype? And this is just a cartoon example. We'll still see real examples in a few minutes in a real case. But conceptually, if we already know that there's a 10 and a 12 that's supposed to be present, and there's now a new 11 allele that we haven't seen before, we have to account for that 11 allele. Also, we're not seeing an 8, a 9, or a 13, or a 15, or a 21, so we have to account for the fact that we're not seeing those either. So if you were to just in the simplest possible make a list of what this could be, well, any possibility would have to include an 11, and it can't include anything except a 10, 11, or a 12. And those are the three possibilities you see on the right. A 10, 11, an 11, 11, and 11, 12. In this simplified world like this, not the real world. Those are the only three possibilities that could be there. And if you're going to make a list and do science, you have to give them some weight. And in science, that's done through probability. Probabilities add up to 1 or 100%. And in this example, suppose those probabilities are 20%, 30%, and 50%. The 11, 12 may have a bit more probability because if you add the 11, 12 to the 10, 12, you would get 2 doses of the 12 allele. So maybe that's a little more probable. Regardless, that's done objectively. That second genotype is now separated out for the second contributor. The first was a 10, 12. The second genotype is this probability distribution over genotypes. How informative is it? Well, you started off with 100 possibilities of allele pairs, and you reduced it down to 3. That's really informative, actually, in the real world. The result would be that when you make a comparison, so now later on a comparison is made to a known genotype that's an 11, 12, the fact that that 11, 12 has 50% probability would simply have the match statistic. If the match statistic was 20 before, it's 10 now. That's qualitatively how it works. But again, we'll get into more detail. So that, and with a broad brush, is DNA mixture interpretation. And let's get into the evidence in this case. So from the right glove, the purple knit glove that the perpetrator was wearing, we're going to look at one of the 15 locations. This location has a name. It's called FGA. And this is what the data look like. We see about 5 peaks here at 20 and 21, 22, 24, 25, 26. There are different heights. Some of them are taller. The 20 on the left is up at 400. The one next to it at 21 is down under 100. And each peak that you're seeing where the allele is labeled on top inside the box gives you information. It gives you the information about which allele it is. That's the size information. By the way, the numbers on the bottom is the size of the paragraph. How long is this paragraph? Like 200 or 250. How long is the paragraph? The number of repeats or the alleles figured out from that genetic paragraph size. And peak height indicates roughly a relative quantity of DNA. The units on the vertical y-axis is RFU, which is the relative fluorescence units. It doesn't tell you the exact quantity, but what you can see here is there's a bunch of alleles present. It indicates that there may be 3 or 4 people because there's so many alleles. You have to account for that. It indicates relative quantities, which is important if you're going to use all the data. It also has a pattern. We see taller alleles on the left and the right at 20 and 26, shorter ones in the middle. That pattern information can be used not only for separating out genotypes, but for also letting computers measure how much variation there is. The starting point for any analysis when you use Bayesian statistical computing is the data. There isn't a concept of good data or bad data. That doesn't mean anything in this type of computing. The data are the data. Whatever you have, you have. And now you have a model, and a model that gets very complex, that models uncertainty and many variables like stutter and relative amplification, different genotypes, mixture weights, quantities, and so on. You may have 100 variables. If the patterns that you see are really easy, then your model can be very specific about what the possibilities are and be very precise and produce sharper information. If your data is more diffuse with lots of variation, all that means is there's more variation and there's more possibilities. If you reach the point where everything is present and it's all random, you're going to end up with no information at all. But the data are the data. And anything that manual analysis would call an artifact or something bad is just another set of variables that are entered in order to model the data. So our starting point are the data, and now let's see what a computer can do with it. What people can do with something this complex is basically try to simplify the problem in order to get a statistic like an inclusion statistic, probability of inclusion at one locus, then combine it across multiple loci. And the way this is done is by using thresholds that sort of mimic what statistical variables will do. There can be a lower threshold that describes the noise characteristics of the machine and detection of things that may not be alleles. There can be taller thresholds, a second one called a stochastic threshold that's trying to understand the counting process where the variance is changing. But whatever it is, one or two lines are drawn, and when that's done, all this quantitative pattern is reduced to a list of all or none events. The highs go away. They're just turned into this allele is present. The lows go away. It's turned into this allele is absent. And by reducing highly informative patterns to qualitative all or none events, that leads to a loss of information, as we saw in this case, to the point where the lab in New York couldn't call it at all. What computers do if they try to solve problems like this is they start with a rich model that includes, as I said, genotypes, stutter, variation. Each variable has its own uncertainty attached to it. But let's make it much simpler now and only look at allele pairs from individuals and how much is present and see what they add up. Let's just focus on a few of the main variables. What we're going to try to do then is generate a pattern, the computer's doing this, and see how well that pattern compares in terms of the heights of rectangles with the data underneath it. Suppose there's one person's allele pair shown in blue from like a major contributor that may be over half of the DNA. That's a 20-26 allele pair, so that's shown in blue. And maybe there's a second person's allele pair, a much smaller amount, maybe only 15% of the data. Then that's in orange, and that allele pair is a 21 and a 24. We see that in orange. And now a third person's allele pair we could propose would be a 24 and a 25, and that's shown in green. If we add up the quantities of those different alleles, then we get a pattern of high, low, nothing, nothing, intermediate, intermediate, and high. The heights of those added up together rectangles are close to the heights of the peaks. Now, in fact, in this particular example, it wouldn't hurt to assume a fourth individual to try to cover that 22 peak and a very trace amount. And we had the computer do that as well. What's interesting is that in this particular pattern, the major blue contributor actually does correspond to the victim, the orange. Minor contributor does correspond to the elimination sample, and the green will end up corresponding to the person who will end up to be the suspect. The computer doesn't really know or care about any of that at this point. What it's doing is it's going to try out thousands in a quick computer run like we did the first day, or tens of thousands, or even 100,000 different possibilities. It's going to shuffle all those rectangles around. It's going to have large amounts of blue or green, or smaller amounts, different amounts, and move them every place. Some patterns will explain the data well here, and that will confer a higher probability to each of those contributors, these three different contributors at this locus. Other patterns, like if you take the 20 and you shrink it and move it all the way to the left, well, now you're trying to explain nothing, and you're not explaining the data that's there. That'll have lower probability. So by trying lots and lots of explanations, the computer can separate out the genotypes of the 3 contributors, or 4, depending on how you're running the problem. So for each of the 15 locations, for each of the 3 or 4 contributors, you will end up with a genotype. And this is what the genotype looks like for FGA, for the second contributor. Instead of having 100 possible allele pairs, it's been reduced to maybe, I don't know, 15 or 20 allele pairs. There's been some concentration of probability based on what the data is trying to tell us. Some probabilities that you're not seeing here have been reduced because the data has made them less probable. Others have been increased. The process is done objectively from the data. It's not looking at the defendant in particular. It just doesn't see a reference. So you get this objective separation into 3 or 4 different contributors at every locus. And this is what a genotype looks like for evidence. In order to get a match statistic, we have to compare evidence with reference or another evidence relative to a random population. That's the math behind match statistics. And so now that we have the evidence, let's see what the random and the reference genotypes look like. The question we're trying to answer is how much more does the suspect match the evidence than a random person? So we still have the same evidence distribution in blue. That's the same that we had before. But now there's also next to it a random population genotype from a randomly constructed person from these population databases that governments and companies create. And that's in brown. The first thing you see is the blue pattern and the brown pattern are different. So we know there's information here. If we look in the very center of the picture where it says allele pair, and we look at the 22-24 allele pair, we see that the blue and the brown bar have the same height. Blue is what we saw after seeing the data from the evidence. Brown is coincidence from before the evidence. If that were the person's genotype, who we were comparing with, because they're of equal height, there's no net information. The ratio is 1. On the other hand, if we look to the left, we go over 1 to the left, and we look at the 21-24 genotype, that would be an exclusionary number because the blue bar, looking at an evidence match, its probability is maybe a quarter of the brown of a coincidental match. So the result of this process can give you an exclusionary number or an inclusionary number or an inconclusive number. In this case, it turns out that Casey Wilson's genotype was a 24-25. That blue bar has a probability for the separated genotype of around 30%. The initial probability of a random person in the population is around 3.5%. And that ratio of the probability of an evidence match, shown in blue for around 30%, divided by the probability of a coincidental match of what a random person would have, around 3.5%, is 8. And that's what a match statistic is. All valid match statistics are ratios of 2 probabilities. The probability of an evidence match divided by the probability of a coincidental match. For example, random match probability with single source DNA is just like this, but the blue bar is up at 100%. So that would be no uncertainty about a match divided by a small amount of coincidence, giving a big number. When there is uncertainty in the data, there's more than one possibility. It all works the same, but now that numerator, the probability of an evidence match, shrinks based on whatever the random factors are in the data. You're still dividing by the rarity or the coincidence, and you're getting a match statistic. So in forensics at this point, computationally, there's no such thing really as a match. There's a match statistic. And the match statistic is the ratio of 2 probabilities. The probability of an evidence match divided by the probability of a coincidence. This can be done and is done across 15 different locations. And when that happens, the x-axis is showing the numbers on a scale about from 1 to 10 for the 15 loci. They're independent genetic sites, and when they're multiplied together, we get a number that answers the question, is the suspect in the evidence? And a way to state that as a ratio of 2 probabilities is that a match between the glove and Casey Wilson, that's one event, is 30 million times more probable than a coincidental match, in this case to an unrelated black person. The reason the stats are listed 3 times is, in this case, there are 3 different databases. New York State called them Black, Caucasian, and Hispanic, and I'm showing you the smallest number. When you're saying that something is so many times more probable than, you're talking about the probability of that match between the glove and Casey Wilson relative to the probability of a coincidence. This is the plainest English you can get the math into. Basically, languages of match, probability, and items. In this case, there was a left glove and a right glove, and in separating out those genotypes and then afterwards making comparisons, we see that the victim and Casey Wilson are both in there with match statistics in quadrillions, thousands respectively. The elimination is really around 1, can't say very much. The right purple knit glove has all 3 people in there based on the match statistics at the levels of trillions, thousands, and millions. So by separating out all the contributors, statements can be made about all of them. This is expressing the same information in terms of the number of zeros in the number. 14 is a 1 followed by 14 zeros, which is around 100 trillion. 4 is around in the tens of thousands, and 7.5 is a 1 followed by 7 zeros, or around 10 million. And scientists will often describe it this way, and often juries can understand it that way, because when you're talking about a trillion, I mean, you sort of get lost with all the numbers. Let's take a look at a report. This is the report in the case. There's a report, and then there's a case packet that goes with it that gives some backup. Here's the first page. Let's take a look at the top half. The top half gives information just about the case numbers, who it's going to, and then it indicates the names of different roles of people: victim, elimination, and suspect. I've changed the names here to victim and elimination and just kept Casey Wilson's name. There's a listing of the items, which can be both evidence mixture items as well as reference samples. Then looking at the middle part of the page, there's a description of the methods. The methods talk about where the data come from. In this case, it was identifier plus from the New York State Police. It talks about applying the TrueAllele casework system to the data and independent replicate runs and what that process is about, and then that match statistics were calculated after the separations were done. And then more specifically, for each item in this match report, there's a preamble that summarizes what happened with a listing of the match statistics in English relative to the different ethnic groups. And to be consistent, let me continue with the one we had before. This is on the next page for the right glove. I'll read it to you, and this is sort of English, but it says this is what happened. The computer assumed that the evidence sample data from the glove, item 18, contained 3 contributors. Separately, we assumed that it contained 4 contributors. We did multiple computer runs, and objectively inferred evidence genotypes solely from these data, meaning it wasn't comparing anything to the suspect. In some of these runs, after we established the victim was in there, we could use the victim's genotype. After we established an elimination sample was in there, we could use him as well. And so we could peel away this mixture to get at the unknown that we wanted to extract as a genotype and make a comparison with. Following that genotype separation inference, the computer did compare a genotype from this evidence to the provided reference items in order to compute match statistics. They're just match statistics, but technically they're called likelihood ratios. They're those ratios of probabilities which all match statistics are. And then we could see in English, this is for each of the 3 individuals, for the victim, for the elimination, and for Casey Wilson. We can read from the bottom that a match between the glove and Casey Wilson is around 3 million times more probable than a coincidence. So that's what's in the report and why it's there. You've already seen these tables where we list evidence items in the rows and the columns have reference samples. with the numbers written out as well as expressed in logarithmic terms as the number of zeros after the 1, or the orders of magnitude. And then the last page, page 4 of this particular report, talks more about the method. I won't go into it in any great detail, but it says what the method is. It talks about the concept of having 2 steps, one of separating a mixture into genotypes, and then a second step of making comparisons of those separated genotypes to different references. Really, what's the point of doing all the separation? If you just have one contributor, life's pretty easy, right? You're comparing one genotype from single source to one genotype to a reference relative to random. With a mixture, it's all mixed in there together. So the philosophy is to go back to that easy problem we had in the first place. Just separate it out into the, say, 4 different contributors in the mixture, and now the comparison is being made from one contributor's genotype from that mixture to a reference relative to random. That's almost the key idea. If you can separate out a mixture into its component parts, you've made it just as easy as it was 20 years ago before you started looking at mixtures, because you're comparing one genotype to one genotype relative to random. We also have references in the bottom section, validation papers. The most important thing you can do in science is to show that your systems and methods work by testing them. You can test them with real data in forensics in 2 ways. The first 4 papers that we published, and actually the 4th one is published by an independent group in Virginia, is testing the TrueAllele system on samples of known composition that are made in the laboratory. So we kind of know what the answers are supposed to be in terms of who's there and who's not there. And these papers go out to 5 unknown contributors. The first one and the third one are online because they're open access. The last 2 here are also open access. So you can read through these. But what they show is what is the false positive rate, the false negative rate, the reproducibility under different conditions of high amounts of DNA, low amounts of DNA for 2, 3, 4, and 5 contributors. But the other 3 papers talk about casework samples, where the data are obtained from casework, from sexual assaults, property crimes, homicides, whatever you have. As the papers move along, the cases get increasingly difficult. The first one was mainly 2-person mixtures. By the time we get to the last one, it's a lot of 4-person mixtures as well. And there again, we're looking at the same sort of metrics. Sensitivity, which is not getting false positives. Sensitivity, which is not detecting what's there. It's really the easiest way of saying it, as opposed to going to a triple negative. Specificity, the extent to which you're not getting false positives, and reproducibility. Reproducibility has been an issue with mixers in the forensics community, certainly since some studies 10 years ago showed that on the same mixture, different groups could get completely different numbers ranging over 10 orders of magnitude on the same data. And I'm going to end by just showing you, because I think I have 5 minutes left on your clock, right? What a case packet looks like. So in addition to having a short report, we also provide something that's over 100 pages long. And I'm not going to go through all 100 pages, but this is just some of the highlights. This is the table of contents. And if you look at it backwards from the bottom up, it explains what we have. We want to justify the matches that are being reported. And we can do that by showing tables of different types and how specific matches are. But a match statistic comes from combining 3 genotypes. And here they are: evidence, reference, and population. So we describe those 3 genotypes, the evidence ones that have been computed, population databases, and so on. How do we get those evidence genotypes? We request the computer to do things. And so here are the requests that are listed out with their timings. What does the request know what to operate on? That's the data. And we list the data in table form as well as in electropherogram or EPG pictures. And our case notes describe what we got. Now going forward, these are very detailed case notes that in a standardized way record our observations about the data and what we did. This is what electropherogram data looks like in TrueAllele for each of the different loci. These are the different requests that were run on this case for the 2 different items. assuming 3 or 4 contributors, as well as looking at the gloves jointly together, which you can do with a computer. Just look at all the data simultaneously. This is what the genotypes look like in tabular form instead of as a bar chart, where the allele pairs are listed with the probabilities. And this is what the match statistic looks like at every locus, which was that other bar chart that we looked at. This is on a logarithmic scale. You add up all the powers of 10, you get a number like 7.5, take 10 to that power, and it's 30 million. Notice that these are all positive numbers, meaning that there's inclusionary support for Casey Wilson being in the glove. If there are negatives, that's okay too. The numbers can go both ways. This picture shows that if the specificity of the result, most people would be excluded with a match statistic of around 1 in a billion from this data. This is a scale of the powers of 10. You compare against thousands of random genotypes, and that's what this room looks like if we compared your genotypes. You're not in that glove, but Casey Wilson's over here as an outlier at around 7 or 8 log units. He's in the glove. So the outcome was that Mr. Wilson was sentenced in the rapes and getting concurrent 20-year sentences. If you want to know more, there are a lot of YouTube talks and papers and all kinds of things on our website. These are 3 general ones that I gave this year. This was a 2-hour CLE at the courthouse that I gave in February that covered challenging DNA evidence from the perspective of the evidence and how do you challenge it. This was a short 20-minute talk I gave at a National Institute of Standards and Technology conference, or NIST conference, that is also designed for lawyers and the general public to get a sense about relevance, reliability, and acceptance of mixture interpretation. And the last one was a talk I gave a few weeks ago to PACDIL, as a CLE, Pennsylvania Association of Criminal Defense Lawyers, of different strategies of how do you actually ask about the reliability and the acceptability of DNA evidence. If you don't want to just watch movies or read about it, but you actually want to do it, you can. TrueAllele used to be these server computers that filled up a room. We still have those in our office down in Oakland. We can also put it on a desktop or in a server room. But what if you don't want to buy a computer, but you have a laptop and you've read a lot, you've watched all the movies, you have the t-shirts, right? And you want to run it yourself? Well, you can. And so TrueAllele now lives on internet, and you can do it from Starbucks. If you'd like, we can get you set up. We use this with crime laboratories who have the system for additional training, validation studies, if they need spare capacity that they want to rent instead of buy. We can use it for solving various interesting cases by prosecutors or police. It's also very useful for the defense. Suppose you have TrueAllele evidence and you want to ask, is this reliable? Well, just run it yourself and take a look, and you'll see. I mean, okay, we ran it 6 times. You can run it 6 times yourself and take a look. It's also good in education, and we use that in the training classes that we have for different universities. I think we're having another one coming up in a few weeks for universities. So there's more information on our website with courses, presentations, webinars. I mean, just allocate your whole summer to this and you probably still won't get through it. But that is what DNA match is. It's a ratio of 2 probabilities of evidence to coincidence, and that's what match reports look like done by computer. Thank you very much.